Okay, we'll finish up chapter 5. We need to talk about sequencing and how if we know the arrangement of amino acids, that can be useful. So, first of all, sequences of proteins are needed to determine the three-dimensional structure, which we'll talk about more in the next chapter. It allows us to make good guesses about the structure and function by comparison to known proteins. So as we start to collect more and more of these sequences, we get greater insight into the structure and function of unknown proteins. And it can also help us find and address disease-causing mutations. So if we look at early protein sequencing, the early efforts were a lot like sequencing DNA. Now in DNA, you only had 4 bases, so there was some greater simplicity there. With proteins, you've got up to 20 amino acids, and you've got the complexity of disulfide bonds. It took Frederick Sanger about 10 years to figure out the structure for insulin, and that's 51 amino acids. So how his method worked back then was he would hydrolyze the protein into chunks, different parts. And then he uses dinitrophenolbenzene, and it reacts with the amine, and then treat it with HF, and it would hydrolyze the peptide, and so you'd get smaller fragments. And then he would take each of those yellow fragments, so DNFB is actually a yellow compound, so he would separate them by chromatography or some other method, and then he would figure out what that little chunk was. And so the key to this is that you had to do this, it was very repetitive, not that you do the same sequence twice, but you'd have to do a bunch of different chunks, and you'd end up with overlapping sequences. So this idea of sequencing overlapping sequences is still used in sequencing proteins. So if you have a big polypeptide, in fact, even one that's got 2 strands connected by a disulfide, first thing is you would reduce the disulfide and you'd end up with individual strands here. And then you take those strands and you'd either chemically or enzymatically break them into smaller pieces. And then each one of these pieces you would have to identify and characterize. And then what you do is you would start to align. And so you see this cysteine, tyrosine here, and you realize, oh, that is the same fragment there. So the cysteine tyrosine has a phenylalanine and a lysine on either end. And oh, this strand right here, this glycine valine, anyway, that ends in arginine. Oh, look, we've got this same one here. Oh, so we've got a phenylalanine here. Well, maybe this phenylalanine is that same phenylalanine, and then it goes cysteine, tyrosine, lysine. Anyway, we'll see another example later, but what you do is you just take all these little individual fragments and you'd start to align them. And if you get enough of these little fragments and enough alignment, then you can actually get the entire sequence. But you can see that it's somewhat redundant because this piece right here actually doesn't give you much information, but it took work to get that sequence because that's actually embedded here. So we'll talk more about that, but that is part of the process. We can see further those alignments. So you take all the sequences that you know, and you would start to align them. And you can see how this phenylalanine here kind of bridges from this strand and includes this strand and then includes this. This is a very simplified version. You can imagine that if this was 100 amino acids long, just how much work you'd have to do to get those individual little fragments. And again, kind of this arginine here is the one that's bridging, And so really, if you're reading, you'd come along here, and then you're like, oh, go down to arginine there. So really, these fragments here don't give you a lot of information, but they are important. And one issue that you have is longer fragments are harder to sequence, and you may have errors in there. So let's say that you actually sequenced this one here, and you got an error. This could be a correction down there. So the more overlap that you have, even though it may be redundant, the more confidence you have in the sequence. So another way to determine the amino acid is by just modifying the N-terminus. And so you can use a fluorescent dye such as danzyl chloride here. And so you would react it under mildly basic conditions. And then after treating it with aqueous acid at high temperatures, you would hydrolyze those peptide bonds. And so you could actually go and isolate this fluorescent fragment, and that would tell you which amino acid was at the end. And after a lot of work and other methods, if you could just march down here and do one amino acid at a time, that would be very advantageous. But you can see right now I've got a bunch of individual amino acids in there, and so it'd be hard to figure out what the next amino acid is. I figured out one, but let's say we've got 99 more to go. So we're going to talk about ways to get around that. So it's important to know the sequence of the protein. Now, they've advanced it where you can actually get the sequence from the DNA, especially with DNA technology and overexpressing proteins in other organisms. That comes directly from the DNA. But if you want to just take an unknown protein out of some organism, there's still methods used to determine that sequence. And one of those methods is Edman degradation, which is a classical method and actually relies on the two chemical methods that we just talked about, kind of this N-terminal sequencing and breaking the polypeptide up into smaller chunks. And so the key here is that what we do is we do successive rounds of N-terminal modification, and then we cleave off that N-terminal specifically, identify it, and then we'll have a new N-terminal because we've removed that N-terminal, and we just kind of walk our way down the protein. So we'll see that in just a second. Now, this modern method, you can actually sequence by mass spectrometry using some very advanced instrumentation, one of them being a MALDI-MS and electrospray ionization mass spectrometry, as well as what's known as tandem mass spectrometry, or MSMS. So now if you have a protein of interest, you can often just sequence it using instrumental methods without doing some chemical modification. Again, those are the things that we'll talk about. So in step one of protein sequencing, you need to break disulfide bonds, and these disulfide bonds are either within the same strand or between strands. If we're going to sequence, that just adds some complexity there. So we want to get rid of these disulfide bonds. And so what you do is you take actually another thiol, and this thiol essentially will come in here. You get a mixed disulfide bond, and eventually you'll reduce down this to the cysteine to free thiols. And what you've done is kind of a dithiol exchange, or a disulfide exchange, sorry. So you now have a disulfide in between your protein. You throw in a bunch of this stuff here, and you've exchanged the disulfide for this smaller compound that you wash away. A more common one that's used now is dithylthiritol, DTT. It's very effective at reducing disulfides and stinks a lot less than mercaptoethanol and other methods. And one of the advantages here is the disulfide that it forms is actually this cyclic structure right here. So if you have a disulfide up here, this example here is just between two side chains, and so you throw in this DTT, it comes in and attacks. Oh, let's get the right one. Attacks here, kicks those electrons onto that sulfur there. This can get protonated, and so now what you've done is made a disulfide here between the DTT and this protein. Now you just happen to have another free thiol within the same molecule, and what it'll do is it will wrap up and attack itself and kick those electrons back to the protein, and you end up getting the disulfide molecule here, and you've got two free thiols. So DTT is actually really cool in that it will form a disulfide within the same molecule there. Now, you reduce these down and get these free thiols, but the problem is they will go and form disulfides again in the presence of oxygen and other reagents. So you need to cap those cysteine residues. They're problematic, they can get oxidized, and so they're very good nucleophiles. And so once you cleave them, then you just go and mix them with some reagent that they react with. And a very good reagent is iodoacetate. And having that iodine on there makes a good leaving group, so that thiol will come in and attack that carbon and kick out the iodine there. And then what you've done is made a sulfide with this carboxy methyl group on it. And that's going to be a unique amino acid residue now, and so you'll be able to identify it later. So anyway, sulfurs are very reactive. So what we've got to do again is get rid of that the disulfide and then modify them. So down here at the bottom is a picture of insulin. And so one of the first one to be sequenced, only 51 residue, took about 10 years, 100 grams of protein. And one of the first steps was to break those disulfides and modify them so that they didn't form again. Okay, so that's step one. Break the disulfides and modify them so they don't reform. Step two, you've got to cleave the peptide into smaller pieces. Now, if you just used a hydrolysis reaction, you're going to get a bunch of random sequences. And that's what Sanger did, and so that's one of the reasons it took so long, is they were just random. So if you could find a way to specifically cleave at different sites, you would get more defined fragments. And so there are enzymes that end up cleaving peptides at specific spots. There are exopeptidases that actually remove terminal residues. So they start chomping on the end and work their way in. And then there's endopeptidases. And they catalyze the hydrolysis of amide bonds. And some of them can be very specific, such as trypsin here, which will cleave on the C-terminus side following a lysine or arginine. So if you do a tryptic digest or use trypsin, wherever you have a lysine or an arginine, it will cleave. And so you can imagine you're going to get very defined fragments because it's only cleaving after lysine or arginine. And what you can do is actually a partial digestion. So you don't let the trypsin cleave after every lysine or arginine. You let it kind of hit. And so it may get only every third lysine or arginine. And if you do this right, then you just get these random fragments. And I'll show you why you want kind of these random overlapping fragments in a second. But that is one advantage that, or one technique that we can use. And so here are some other enzymes, some various endopeptidases, and some of them have more specific cutting or hydrolysis of amide bonds than others. So there's just a little schematic here. If this is the bond of interest here, what we refer to, this is the residue that's N-1. So it's just up one. And here is residue N. doesn't really matter the nomenclature. This will just kind of let you know how things work. So trypsin, and notice we're drawing it from the N terminus to the C terminus. So trypsin, we get it from the bovine pancreas, and one reason you'd want to do that is we end up slaughtering lots of cows, and so you can isolate this from the leftovers after getting your hamburger. And so what they say is this residue that's N-1 needs to be a positively charged residue, such as arginine and lysine. And then the following residue can't be proline. So that's how you read this. So chymotrypsin cleaves after bulky hydrophobic residues, such as phenylalanine, tryptophan, tyrosine. Again, that needs to be in this position. So if you have one of those bulky hydrophobic residues, then you'll cleave that bond as long as this is not proline. And one of the reasons that you don't want proline, or it doesn't work with proline, is remember proline actually back bonds and forms a cyclic structure there. So if you have the proline there, it makes it more difficult to cleave that amide bond. And so you see most of these won't cleave when the following residue is proline. So here's another one that cleaves after small neutral residues like alanine, glycine, serine, valine. Again, you can't have proline at that position. Now this one here, RN. So this one right here, if it's isoleucine, methionine, phenylalanine, tryptophan, tyrosine, valine, it will cleave, but this RN-1 can't be proline. So you don't need to memorize these, just understand how to read this chart. You know, if we gave this on the test and then I gave you a certain amino acids, and you could figure out where it would cleave, but this information would be provided for you. You probably should know trypsin and chymotrypsin. They're kind of the ones that you run into if you have a standardized test. Trypsin cleaves after the positively charged residue arginine, lysine, and chymotrypsin cleaves after hydrophobic or the kind of aromatic residues. So that's probably good to know just for general knowledge, but really what we want to be able to do is read this chart, given a sequence, be able to identify if that bond would cleave or not. Now there are some chemical reagents that will also cleave. So cyanogen bromide, we're going to talk about that one. That cleaves on the carboxyl side of a methionine residue, but there are others here. But we're going to focus on cyanogen bromide. And again, here's just some other enzymes. So you just need a way to directly cleave. So let's talk about the cyanogen bromide. So it reacts specifically with methionine, and here is a mechanism. So that sulfide, that sulfur in methionine, will attack cyanogen bromide, which is the structure seen here. Now it's attacking a sp carbon, so kind of an interesting reaction, and it kicks off the bromide. I don't believe this is the real mechanism because those orbitals won't align. It makes it look almost like an SN2. I believe actually the electrons would come in and attack that carbon, kick them out onto the nitrogen, and the nitrogen comes back and kicks off the bromine there. But we're in biochemistry, so this is good enough for us. Now the whole point is what we've done is we've now modified that sulfur. Notice it's positively charged, but we've modified it with a nitrile or a cyanide group, a C triple bond N. And that sulfur ends up being a good leaving group because it wants its electrons back. And so we'll talk about this more next chapter, but there's a significant amount of resonance in amide bonds. And so this nitrogen following the methionine residue can donate its electrons in, and that will push those electrons from the oxygen, the carbonyl, up and kick this out. So really what you've done is just made the side chain into a good leaving group and allow this cyclization to occur. Now, after that cyclization, you end up with this group right here. And hopefully from organic chemistry, you can identify that group as an imine. And it's a derivative of carbonyls. and in the presence of water will be hydrolyzed back to a carbonyl. And once you hydrolyze that carbonyl, you'll get your free amine and the carbonyl back. And so here is your methionine residue. It's been modified greatly. In fact, you've lost the sulfur. This is called homoserine. And it's a homoserine because it has this oxygen here. And the term homo in naming structures means that you actually have one additional carbon in there. So there are structures that are known as homocysteine, and that would have two carbons in the side chain. Or if you had homoalanine, or no, not homoalanine, homolysine. I don't know if that even exists. Hey, that'll be our bonus questions. Go and figure out if homolysine exists. But homolysine, if it does exist, would actually have 5 carbons and then the amine. So for one bonus point, figure out if there is a homolysine. But again, the term homo, just in this case, we use it in chemistry to mean highest occupied molecular orbital. But in naming structures, it is just a homolog, I believe, is where the term comes from. And that just means that the structure has one additional carbon than the parent name. So this has one additional carbon than serine. Anyway, side note there, but thought you ought to know where that comes from. But yeah, tell me if there is such thing as a homolysine. And we'll make this a double bonus point. Provide the prefix or the term if something has one carbon less. There is a term that you append. So if you had lysine that only had 3 carbons, that amino acid I know does exist. It's known as ornithine, but there is a prefix. You would say something lysine, and it means that it has 1 carbon less. And then if you said homolysine, it has 1 more carbon. So there you go. 2 bonus points for this one. Tell me if homolysine exists and what is the prefix or the term that you append if something has 1 carbon less in naming. Okay. So I switched quick. So I'm actually going to go back a little bit here. So we've got a couple different ways to cleave specifically polypeptide bonds, and that's really important. Okay, so we can do enzymatic, or we can do chemical. The one chemical method that we need to know is this cyanogen bromide, and it cleaves after methionine residues. So once we have these smaller fragments, what was discovered was Edmond came up with a way to sequence. Now, this is just really super clever. Took advantage of the N-terminus and its nucleophilicity, and it reacts with phenyl isothiocyanate, also known as PIT or PITC. And you can see the mechanism here as it attacks, it kicks those electrons onto the nitrogen, that gets protonated, and you end up with this functional group here, known as a thiourea. And so you can see in blue was the N-terminus of the amino acid, and you can see the group that you've now added. So this is known as a PTC polypeptide. Now, if you treat it with anhydrous trifluoroacetic acid, it's a fairly strong acid. So it's acetic acid that has 3 fluorines here. Those 3 fluorines pull away and make that acetic acid much more acidic than just regular acetic acid. If you treat this this modified polypeptide with the trifluoroacetic acid, what happens is you get a arrangement and you get this thiazolione derivative. And so you can see kind of how that mechanism, again, the nitrogen here is very good at donating its electrons in, kicks those electrons out to the sulfur, and the sulfur goes down and attacks the carbonyl. Again, biochemists, they kind of miss out on some of the steps of the mechanism, would kick up onto the oxygen, collapse down, and then we would break that bond. So the key is we get this cyclization from the sulfur to that carbon, and we cleave here. Notice the ingenuity of this. We actually just break off one amino acid. That blue peptide, or that blue amino acid residue, is broken off of the peptide, and we actually end up with this peptide intact, just with one less nitrogen, or sorry, one less amino acid. Now, this thiazolione derivative actually undergoes a rearrangement in acidic conditions to this much more stable, referred to as a PTH amino acid. Now, the key here is this part in red came from the derivatizing agent, and this thing right here came from the amino acid. So each amino acid is going to give you a different structure here. So you'd actually end up with 20 different PTH amino acids. And then these come off and you purify them, and each one of these has some characteristic properties because that R group side chain is going to change. So what you can do is just do this stepwise, and you can identify those PTH amino acids, and then you know what the N-terminus was. So here's just another way of looking at it. So you take a protein, you want to, if there's multiple of them or disulfides, you break them into the polypeptide. You often want them in smaller chunks. So this Edman N-terminal sequencing actually will sequence reliably up to 100 amino acids. And so what you do is derivatize it. That cleaves off the N-terminus. You lower the pH, and this comes off, and this stable structure right there is what's analyzed. You recapture the rest of your polypeptide, and after you've purified this out and identified it, you go and run this through again, and then it would cleave off another N-terminal residue. You identify that new residue and repeat this over and over again. And it's been automated. Like I said, it's not as good as DNA sequencing that can get up to runs of 1,000, but it can get up to runs of 100. And I just have another picture in here. This one's kind of interesting in that you can see that here's my N-terminal sequencing. I do all this labeling, I release it, I identify what's in the yellow box and retain this segment to sequence again. And then I label it again, release it, and then identify the second one. So lots of manual effort here. You can't just kind of just run this all in one beaker. You've got to do some purifications in between here. But what we end up with is we end up with a bunch of these overlapping sequences. Now, if I just did a complete digestion of a protein, as you can see up here at the very top, you can see that I get 2 alanines, and I get a glycine and 2 lysines, and phenylalanine and threonine and tryptophan and valine. So a complete digestion lets you know the amino acids, but it doesn't let you know anything about the sequence. And like we talked about before, the sequence is very important. So if I take a digestion and I use trypsin, hopefully you remember what that does. It's going to cleave after positively charged amino acids. So at the C-terminus, you'll always have a lysine or an arginine. Chymotrypsin is going to cleave after the hydrophobic aromatic one. So you'll either have a tryptophan, a tyrosine, or a phenylalanine at the C-terminus. And then you've got to sequence these small fragments here. So that's key. So once you sequence those small fragments, what you do is you go in a line. And you can see that, again, you're getting some redundancy here. So these red and blue all came from the same protein. And so what you do is you start saying, oh, look, there's a val lysine here. I wonder if that val lysine overlaps there. And so you lay that val lysine under there, and then you realize that you have an ala-ala trip. Oh, look, this ala-ala trip probably continues there. So the next one after trip is gly and then lysine. And so eventually you put those together and you get the complete sequence. Here's just another one. So up there at the top in the purple box, there's our sequence. You may be saying, well, gosh, if I know the sequence, why do I have to digest it? So imagine that this is a black box. And what you do is you take a sample and do the cyanogen bromide cleavage. So that's done separately. You take some sample and you do the trypsin digest and collect those fragments. And again, you don't do a complete trypsin digest or a complete cyanogen bromide. So like if we look at cyanogen bromide, where was that one? Yeah, maybe it cleaves after one, but it doesn't cleave after both of them. Or we do a trypsin digest and it doesn't cleave after both of those. It only cleaves after one. And then what we do is we end up with these fragments. So here's a fragment from a trypsin digest. Here's a fragment from a cyanogen bromide. Here's a fragment from a cyanogen bromide. This fragment right here. Oh, I think I messed up. That doesn't come from any digest. I think I should have cut it right there. But in the end, you get enough of these small fragments and you start overlapping them and you end up getting the sequence. So yeah, I tried to make up one here, but yeah, I should have cut that one off there because it'd be after the arginine. This was a cleavage after a methionine. And so, yeah, you cleaved a methionine there, and then the other fragment would have been something like that. Anyway, so yeah, when you cleave, you'd get two different fragments. The whole point here is we end up a bunch of small fragments that we can easily sequence, and then we overlap them, and we can get the entire sequence. So again, a lot of work. Now, in one of the first steps, what we did was we broke the disulfide bond because we didn't want that interfering when we sequence. So another step that you have to do once you figure out the sequence and where the cysteines are, like we saw back in insulin, they had 6 different cysteines, and you want to figure out who's connected to who. So another step that you need to do is actually keep the cysteines intact and then digest. And then when you digest, you're going to get these 2 connected fragments here, and you'd purify them out. And then you'd sequence them individually. So this is now going to be a fragment here with this cysteine, and then you'd sequence this one here, but you isolated this together so you know that those 2 fragments are connected. And since these 2 fragments are connected, you look at your other sequences, find where this was and where this was, and then you can figure out who is connected to who. Now, if you only had 2 cysteines in there and they were in a disulfide bond, you really wouldn't have to worry about this. But like in insulin, it's like, how do we know who was connected to who? So let me just go back to insulin and just show you that. So how do I know that this cysteine here is connected with a different chain and this cysteine here is connected within the same chain? So like I said, first of all, you'd break them up and you'd have all these little chunks and you'd come up with your alignment. Then a separate step is you actually leave the disulfides intact, and then you would cut them up into chunks. And so you can see here, actually, if we cut this up into chunks, you'd still have 2 different disulfides, and one would be connected to a different chain and one would be connected within the same chain. So you can imagine this could get complex. But let's just take this little sample down here and just say, okay, well, we cut this here, and we just happen to be able to cut that fragment out. And then we separate it, we sequence that, and we sequence that. We know this one has a cysteine here, and we know this one has a cysteine, so we know those 2 fragments are connected. And then what you do is look in all your other fragments where these amino acids appear, and you put them in that sequence. You figure out where all these amino acids appear, put them in that sequence, and then you knew, because of this last step, that those two were connected with each other. So in the end, lots of work to sequence proteins. So gosh, it would be nice if there were easier ways. Hey, we can do it with mass spec. And so we talked a little bit about mass spec in organic chemistry. If you're a chemistry major, you've talked a lot more about it in instrumental. But the method that they use in biochemistry is a little bit different than they use in organic chemistry. In organic chemistry, the one that we teach about, at least, is electron impact ionization. So you get lots and lots of fragmentation. Well, here we don't want really lots of fragmentation initially. What you want to do is just charge the protein. And so what they do is you put a sample in and you put it through an incredibly high voltage. And what happens is charge gets stuck on that protein. And then what you do is you put it into a low vacuum. And, well, sorry, you atomize it. You put it into very small droplets, and then you put it into a vacuum. And the solvent evaporates. And as it evaporates, it leaves individual proteins that have charges on them. And since they're charged, just like in regular mass spec, what you can do is you can attract those charges through magnets. And there's a couple different ways that you can figure out their masses, but essentially you have a mass analyzer, and where they hit the detector lets you know the mass to charge ratio. And you don't get much fragmentation here. Now, what's really interesting about these methods is you can detect multiply charged species. So your protein may have multiple charges. And so here down at the bottom, you actually see these kind of regular dispersed patterns here. And from those, you can actually start to figure out what the charge pattern on them. The math is kind of beyond what we're going to do, but since it does have this regular pattern, you can kind of figure out, and the difference between these peaks, you can actually figure out how many charges are on the amino acid. And so if this has a +9 charge here, this is the mass to charge ratio. So this peak here, how I'd determine the true mass is I'd multiply that by 9, and that would give me the mass. And I'd multiply this by 10, and that would give me the mass. I'd multiply that by 11, and that would give me the mass. And I'd multiply that by 12, and that would give me the mass. And so if you notice here, each one of these, if I multiply by its charge, gives me approximately the same mass. And so we can do that to figure out the mass of an overall protein. Now, what's nice is we can collect this mass to charge ratio between 2,000, but the mass of this protein is going to be somewhere up around 16,000. So if we get these multiply charged ions, then it actually, we don't have to have as much range in determining the mass. So you can see, just by going up to 2,000, we can actually determine the mass of a molecule that weighs up to 20,000 as long as it has multiple charges on it. Now, you can even do something even more amazing, and you can do this tandem mass spec, or what's called mass spec. And so what you do is you take a sample and you ionize it, and you're going to get a specifically charged fragment. What you do is you have this ion selector that only lets a given ion of a specific charge through. And then it goes into what's referred to as a collision cell. In this collision cell, you actually get collisions with an inert gas like helium, and it's going to fragment your protein. And then you have a second mass spec that actually detects the weight of those fragments there. Well, we know the mass of each amino acid residue. And so what we could do is actually figure out the difference between here and here, and that will tell us what amino acid we lost in that fragmentation. So let's see a little bit better picture of this. So we've got a little sequence of amino acids here. The entire sequence weighs 621. And as we start to fragment this, in some cases, you're going to lose that N-terminus residue. Not all the time, but in sometimes when you fragment this, this isn't specific in that you just cut the N-terminus off each time. You're going to get random cuts here. But what you do is you take your essentially molecular ion and you look at the fragment that is the next lowest down. Well, that's likely going to be cut from either the N-terminus or the C-terminus. And you figure out that mass and you say, oh, look at that. That is glutamate. And then you look at the next one down from that and you're like, oh, okay, from this fragment to this fragment here is another glutamate. Oh, look at this fragment here is glycine. The difference between this one and this one was methionine, and the difference between this one and this one was arginine. Now, there's lots of computations in this, and they do the same thing. You need to overlay a bunch of sequences. So it's very computationally intensive, but what we need to understand is if we can figure out the difference between these peaks here, you know which amino acid was cleaved off. And if you get enough of these reads, you can start to figure out what the sequence was. And so here is just kind of a summary of that. You inject your sample. It's in a little bit of liquid. That liquid evaporates and you're left with charges around your protein. And you then go and determine the mass of that. And you get these multiply charged, so we can actually figure out the mass of our protein there. So in fact, this one weighs 47,000, but we can actually identify the multiply charged here and get the mass. And you can see, again, we only have to read up to about 2,000. And we can actually figure out the weight of something that's incredibly heavy just based on these multiple charges. Or we could use a mass spec, where first of all, we put it in here and we select. So likely in here, and I didn't mention this before, sorry, but these are individual fragments. So you do the same thing. You chew up your protein into smaller fragments, and then you select one of the fragments of interest to probe further. And so in this experiment here, you're actually going to probe each of these fragments separately. So you're going to pull this fragment in here, and then you're going to fragment. Now, these aren't fragmented by the instrument. They are fragmented specifically using chemical or enzymatic methods, just like we saw in the sequencing earlier. So we have an idea, and these are controlled fragments here. Here, you're going to get all possible fragmentation. And like I said, it's computationally intensive, but if we can make smaller fragments and then fragment them enough, and we take the largest mass and then we take the difference between each one of these, we can actually end up sequencing a protein just by mass spec. And again, this is simple because you just throw this all on the mass spec and let the computer do the work. It's not nearly as intensive as N-terminal sequencing. And then what do you do with all that information? Well, you take all that information and you store it in a data bank. So here's some links to data banks that contain information about proteins and about DNA sequences. These are often publicly available. There are private ones, but people mine this information for all sorts of uses. And so you can actually go there and investigate. And so if you went to the UniProt database, you could look up sequences. Now, if your name's like mine, McKay Stephenson, you can spell my name out in amino acids, and I haven't done this. I should do this. Go in and search and see if there's any proteins that have the sequence MACKAYSTEFFENSEN. That would be interesting. I should do that and see if there's any proteins that have my name in them. And if it did, it would pull up the protein here, what gene it came from, the organism that it was actually isolated from. You can look at domains that we'll talk about in just a second. If they have some information about the function here. And anyway, so tons of information. So as people sequence these, they'll put that information out for the public so the public can use it and mine it. And this is what's, if you've ever wondered what bioinformatics does, this is part of what bioinformatics does. They take the information, either DNA and protein sequences, and they try to find trends and do all sorts of interesting stuff. So one of the things that they like to do is check and see evolutionary connections between proteins. So here are what we some checkpoints, but let's jump into the next one, which is protein evolution. And like I mentioned, once we have these sequences, we can compare them to reveal the evolutionary relationships between proteins. So I won't read this all to you, but here is some of the values of amino acid sequence. So it provides a wealth of information, as well as gives us information about the protein structure, function, and history. And so these are some of the things that we're going to talk about here. But just to let you know, there's lots of value that we can actually get from the sequence. And one thing is comparing the relationship of a protein from several different organisms. So this is cytochrome C, and they've sequenced it from mammals, other vertebrates, insects, fungi, and higher plants. And what they do is what's called a sequence alignment. And you try to find a point where there is an alignment, and you notice here there is a glycine. And so they aligned there, and they just refer to that as 0.1. Now, some proteins have amino acids before that, and so they just call them -1. And then they just start counting from here. But you can see that, yeah, here's a common point that all these seem to align with. And then they just align all the sequences and they color code it. I'll talk about that color code in just a second, but you can see that, yeah, there's some spots that are all the same color, some where there's, lots of variation in the color. This is the same color all the way down. Sometimes we see only one or two that have different colors there. So let me go here and we'll explain what those colors are. And there's different ways of aligning these, but just in this example here, the hydrophilic acid are all indicated by red. And so you come over to this column here and you can see, yeah, most of the proteins have an acidic acid residue there. Here's an interesting one that doesn't. It has a blue. And so it's got a polar uncharged residue there. So that's really kind of interesting that out of all these sequences here, that's the only one that has that. And you can see some spots, like here, you've got all sorts of different stuff going on in there. You've got these hydrophobic residues, you've got some hydrophilic acidic residues, and you've got these polar uncharged residues. Now you don't have hydrophilic basic. That's kind of interesting. I don't see any of those here. Here's one where you've got polar uncharged and some basic residues that pop up. You notice here, these are all basic. And in fact, if you look at, starting here, the sequence is fairly conserved. Now, this is just by color. You'd actually have to read down and see if the amino acid changes or not. Let me see if I can, so here's an example. They're all hydrophobic, but it may go from valine to isoleucine. But sometimes the amino acid is constant down the entire column. So there's lots of information here. Okay, so what we want to do is just talk about it. So I got a different sequence. This is just comparing two, so it doesn't really have the wealth of information here. But what we can do is we can classify the residue as invariant. And as you'd expect, invariant is the same residue at a particular position. So over here, we're just comparing a protein between human and whales, and you can see that there's lots of invariant positions here. You can imagine the more species that you get, you're going to see some variations. But let me just go back here now that we know that term invariant. And I'll even just go back here. So you notice at position 1 here, everyone is invariant with the amino acid that they have at that position. And you can look through here and find other invariant sites. And then you have conservatively substituted residues. So this occurs at a site that probably has less stringent side chain requirements, but they need to accommodate either the same type of amino acid. So again, let me go back and see if we can find some conservatively substituted. And that's what I was trying to illustrate, let's say in this column here. So if you notice, these are all the same color, but it's not all the same amino acid. So it's been substituted, but it's been substituted conservatively, meaning that you get kind of the same type of amino acid, a hydrophobic one. And then finally, the other type of site or substitution that we get is hypervariable, where it really doesn't matter what type of amino acid is there. It doesn't seem to really affect that. And so you can look along here and start to see kind of hypervariable. That's where you're going to see a bunch of these different types of colors in there. This one's really interesting because you go from a basic to an acidic residue, and you've even got some non-polar in there. But you can see those that tolerate. So, if you had the alphabet coming down here, then you'd know that that is a hypervariable site. Now down here at this bottom, this is from a different book, but they have a key here. So identical amino acids, that's what we would call an invariant. Conservative substitutions are, again, conservatively substituted. And non-conservative substitutions, that doesn't necessarily mean that it's hypervariable. That just means that, that we've changed it. Okay, so it's kind of a non-conservative substitution. Hypervariable are going to be those that have lots of different residues there. So I wouldn't say that this is hypervariable. You'd need more sequences to see really if it can tolerate a bunch of different sequences. So this hypervariable is probably better used if you have many, many sequences that you're looking at. This non-conservative is just a subset of this. Well, you're either conservative or non-conservative, or like me, you're right in the middle. But again, it kind of depends on how big the data set, but these are the terms that your book uses, so understand what an invariant is, and a conservatively, or let's just even say a non-conservatively. That means, hey, I'm going from, a dramatically different amino acid. And then hypervariable, like I said, would be if you had many sequences and you start looking and you realize that at that position, you could really throw in any amino acid. So we can sequence these sequence them, and you can align them and look at relationships between proteins. And so what you do is you make an assumption that related organisms evolved from a common ancestor. So it would make sense that the genes that come from those, and thus the proteins, would have similarities. And over time, small differences arise. And if we look at the amount of differences, we could actually figure out how closely related organisms are. Now, there is a big exception, because you can get what's referred to as horizontal gene transfer, and that is not maybe what you think of. Anyway, there's a funny joke. I won't tell you about horizontal gene transfer. We'll tell you about it in the biological sense, and that is from one species to another. And that can occur, and especially microbes do this a lot, and that really messes up this evolutionary analysis, because, like I said, the assumption is that related organisms come from a common ancestor. But if you start shuffling genes from one organism to another, between organisms, that's really going to mess up your analysis. And so you can look at ways to do these alignments. You can do a global and try to match sequences across their entire length, or you can look at local assignments, look for short segments that match in these. So you can do a BLAST search, which is basic local alignment search tool, and look for short sequences, and you can change the size of that. But let's say you want to find 10 amino acids and see how frequently that 10 amino acid sequence appears in every protein out there. Now, the issue here is that we can do a lot of things with DNA, and so we can do substitutions and insertions and deletions. And so when we start playing with the DNA, we'll actually change the protein. And so natures take advantage of the ability to play with DNA and move around sequences. And what we end up with is these domains that I'll talk about in just a second. But once we take these protein sequences, we can actually start to plot out their relationships. So this is again for cytochrome C. The numbers shown there are the number of substitutions per 100 amino acids. And so the longer this branch here, the greater the evolutionary distance. So it took a long time to get from this common ancestor to this point here. Now, the organisms that occurred in this time are lost, essentially. And we don't have a record of them, but we know, you can kind of figure out, okay, well, we had a common ancestor. That's the assumption that's made, and all these changes over time were lost. And then finally we get to this point here. And then we start to break off. And so this common ancestor here, it took 4 amino acid substitutions per 100 to get to this point, and wheat came off. And so you can see between wheat and sunflower, they're related to this, 7.5 to get to wheat and about 10 to get to the sunflower there. So something that's interesting is to get from the fungal cytochrome C to the human cytochrome C, it took about the same number of changes in the amino acids. And so, like it says there, it's not necessarily that we're more advanced evolutionary. It's not like, we've had a lot more mutations to get to where we are. We're just different. So this here doesn't really tell you about the advancement of the organism. It just tells you evolutionary, that to get to the fungus here took about as many as to get to the humans here. Okay, so as I mentioned, how do these things arise, and how do we get differences? Like I said, nature takes advantage of DNA, and so we can, lots of things that can happen in DNA, but you can get a gene duplication. So when you're replicating your DNA, there's some mistake and somehow you get an extra gene in there. Well, what could happen is you could get mutations, and so one of those genes now becomes non-functional, like you see off to the left, or the genes become more specialized. So this one here ends up doing something, so it gets a mutation and it does something that's more specialized versus this one here. Or what happens is one of the genes stays pretty much the same, this one gets mutation, and then this one gets a new function here. Now, why we want to talk about genes is because these genes are eventually going to turn into proteins. Now, these two here came from a common gene, and so they should have some commonalities in there. Now, they may have different functions, but they should have some similarities in their proteins. And then these two would also have some similarities in their proteins. Maybe it's only a few amino acids, and one does one specific task and the other one does another specific task, but it's the same type of reaction. They just act on different substrates. Again, we don't talk a lot about that till next session of biochem, but what we do want to talk about is how we arise at all these different proteins. Here's just another one. You get kind of a duplication in the DNA, and then over time that duplication can lead to different genes that end up leading to different proteins. And so these different proteins, you can end up what is referred to as orthologs, that are homologous proteins with the same function, but they're in different organisms. So often we'll go and look at other organisms and they do the same type of chemistry as us. And so we look at those and compare those proteins, and those are referred to as orthologs. And so again, they're doing the same thing. And then you got paralogs, that are homologous proteins arising from a gene duplication. that are now evolutionarily independent. So let me try that again. Paralogous genes can be found in the same organism or in different organisms. So you can just see the relationship here. So XA and XB or orthologous. So and then XA and YA are paralogous, as well as XA and YB there. Okay. And so why we want to do that again is try to establish the evolutionary relation. So the globin family occurred through gene duplication and divergence, and so we have many globin paralogs. We'll talk more about this in chapter 7, I believe. We'll talk about myoglobin and hemoglobin, talk about fetal hemoglobin, which is really interesting. I'll just a little side note on fetal hemoglobin. If you want to have your baby absorb oxygen, its hemoglobin needs to have a higher affinity for oxygen than the mother's hemoglobin. So when you're in the womb, you actually have a different type of hemoglobin that actually has a higher affinity for oxygen. And so it takes that oxygen and essentially can shift the equilibrium so it can actually get oxygen through the blood exchange from the mother. If it had less affinity, then the baby wouldn't be able to get the oxygen it needs. If it had similar affinity, you wouldn't favor the equilibrium. So the baby actually shifts the equilibrium. Its fetal hemoglobin has a much higher affinity for oxygen. And so it essentially does kind of a Le Chatelier's principle, and you move the oxygen from the mother's lungs through the mother's blood, through the placenta, and into the embryo, into the child, the fetus. There you go. Anyway, so you can actually look at this relationship here, and these globin proteins have different uses. And again, we'll talk more about those in chapter 7. Now, what we do find is that the evolution of proteins occurs at different rates. So one protein, histone, H4, that's used for wrapping DNA, actually hasn't really changed over millions of years. And so we can tell that because you look at different organisms and they have very few substitutions. So such as peas and cows, we can look at the record and they diverge about 1.2 billion years ago. And there's only 2 differences in their amino acid sequences and their conservative substitutions. So what that means is that histone has been evolved, and changes to it lead to very unfavorable consequences. So it's kind of reached its evolutionary end. And if you start changing it and you get a mutation there, it leads to non-viability of the organism. So therefore those mutations aren't passed down. So there are some changes, but they tend to be conservative. There are other proteins, fibrinopeptides, that may not play as, I don't want to say as important of a role, but you can have a lot more variability in there, and the protein will still function in its role. So histone has a very specific role, and it needs a very specific structure. Some of these other ones have specific roles, but their structure can vary quite a bit. And so depending on how much that protein tolerates substitutions, if I put a single change in the amino acid and it shuts down the function of that protein, well, that's likely not going to be passed on to offspring. And so you can look at the evolutionary rate that things are changing, and essentially it gives you an idea of how well substitutions are tolerated there, and meaning that, yeah, the structure can vary quite a bit and still be functional. And then we get the wide variety of proteins. Like I said, you get these gene duplications and duplication of DNA. And so what we find out is that proteins are actually made up of kind of a small subset of domains. So there's so far around 1,000 to 1,400 domains. And 200 of these are most common. And so what we can do is look for these domains. And so if you find domains with greater than 40% sequence identity, then they probably do the same thing. If they're less than 25%, then they probably do something that's somewhat different. And so what you can do is take these domains like Lego bricks and combine them to make a functional protein with an overall different function. And so here is kind of an example. So we got these repeating domains or motifs. And so we'll talk about these cartoons later, but essentially we've got this helice, we've got a turn, we've got another helice, and then we've got this other domain here, and then we've got this other domain. And so when we combine these domains together, we get a new functional protein. And so here's a example. And anyway, we'll talk about these domains later and actually look at them, but we just represent them by shapes here. And you can see that this domain is repeated over and over and over. Then we put a new domain in there, and it's repeated over and over and over. And then this domain's repeated over and over again, and over here. And so you can see that, yeah, we repeat within a protein these common domains, but you can shuffle these domains, and then you're going to give rise to a new protein. And the thought is that the domains arose because they did something. They were functional. So like we said, you're not going to get all possible amino acid combinations, but if you find one that works, then why not replicate that domain and start putting it together? And if you keep assembling these domains, then you can start accessing functional proteins that may do something different. Okay, that is enough for protein evolution. Just understand that sequencing proteins allows us to understand the relationship between proteins. And as we move into the next chapters, we'll refer back to some of those topics like domains and understand why looking at a domain and understanding what the domains are valuable in trying to determine what a protein function is, which is really what we focus on in the next couple chapters.